This is Dr. Dan Martin for the authors of Pathogenesis of Endometriosis, the genetic-epigenetic theory. Endometriosis is a disease with hereditary, inflammatory, immunologic, and biochemical components. It is also likely to be more than one disease with major late phenotypes that include typical, cystic ovarian, and deep infiltrating endometriosis. The components of theories have included the cell of origin, the method of dissemination, and the transition. The genetic-epigenetic theory is a transition theory based on the changes that occur as an original endometrial or other stem cell is transformed into or replaced by endometrioid cells. It is not dependent on theories of the cell of origin or the method of dissemination. With respect to cell of origin and dissemination theories, no single theory explains all clinical observations, including far distal locations, endometriosis in women with Mullerian agenesis, or endometriosis in men. Of the cell of origin theories, endometrial cells from retrograde menstruation or hematogenous dissemination, in addition to peritoneal stem cells, are the leading candidates in most cases. However, Mullerian remnants are a likely cause in the 17 men with pelvic and lower abdominal endometriosis reported since 1971, and a similarly small number in women. The transition into an endometrioid cell with the subsequent development of typical cystic ovarian and deep endometriotic lesions include a variable series of additional changes in cells which already have hereditary traits that predispose to endometriosis. In addition, multifocal monoclonal lesions in one woman may derive from different progenitor cells. Based on human and animal studies, these changes can progress or regress spontaneously in a non-linear fashion. And based on the age at observation, these small lesions that escape the immune system and other clearance mechanisms can progress through a series of other phenotypic appearances, including typical, cystic ovarian, and deep endometriosis. Furthermore, more than 20 overlapping laparoscopic phenotypes have been described. It is not known how many of those have significant immunologic, biochemical, epigenetic, or genetic differences. Early microscopic and subtle lesions will likely have some normal endometrial characteristics until those are replaced by endometrioid phenotypes. Most early lesions will be cleared by immunosurveillance and other clearance mechanisms, and will likely have few or no significant symptoms in most women. However, some of those lesions with additional genetic or epigenetic changes will develop into the late forms of endometriosis. A consequence of the changes is that typical, cystic ovarian, and deep endometriosis are three different diseases. In addition, since each lesion has been exposed to a different set of stimuli, the lesions are heterogeneous, and this can explain the variable response to medical therapy. As Donna Vogel observed at an NIH conference, studying endometriosis is like nailing jello to a tree. We hope that our article will help you in your quest to understand this enigmatic and distressing disease. Thank you.